So our next speaker is Dr. K.M. Sharika. She is a professor in the Department of Cognitive Science at IIT Kanpur. Professor Sharika completed bachelor's degree in biochemistry from University of Delhi. Then she obtained her master's and PhD in neuroscience from National Brain Research Center, Gurgaon. And after completing her PhD, she went to do a postdoc at Duke University. And before joining IIT Kanpur, she was a research associate at University of Pennsylvania, USA. And today she will be talking about social cognition, a peek through a multimodal lens. Now, let's warmly welcome her to commence the talk.
Hi everyone, you can hear me fine. Thank you so much for the introduction. I'm really excited to be talking to you all, a very unique group of audience. And I thank the organizing committee and the team for making this possible, therefore. And I know it's been a long day for all of you, and some of you might be tired. So I'll try to go through, I'll try to make this light and easy. Not maybe go into a lot of details, but if you have any questions, if something is unclear, please feel free to stop and ask me, okay? So with that, let me begin. So this is the title of my talk, Social Cognition, a Peek Through a Multimodal Lens. I have tried to organize this into broad sections that gives you a flavor of the field and also highlight some of the approaches that we take in addressing some of its key questions. Okay, so why social cognition is important at all? So according to the social brain hypothesis, the primate brain size involved mainly to address the everyday complexity of dealing with social interactions. And that's why we find in many primate orders or families of species, there is an increase in the neocortex ratio, which is the ratio of the neocortex to the rest of the brain. And neocortex is that region of the brain which is typically associated with higher cognitive functions like planning, decision making, evaluative thinking, things like that. So the neocortex ratio was found to increase with the size of the increase in the group size of these animals, and linearly so, significantly correlating like this, as opposed to, say, the fruit content in their diet or the size of their range, which is the geographical habitat that they usually reside in. So where you can see, where you can see the correlation is not so linear, right? So this was to rule out these alternative hypotheses that could have potentially contributed to primate evolution, brain size evolution, which was the foraging theory, which said that it's the foraging habitat, foraging habits of these animals that led to greater brains or bigger brains, or it could be the spatial navigation abilities. If it were the case, then you would have seen a clear correlation with the range size, which was also not so. So it ended up being the group size of these animals that was found to be significantly correlated with the neocortex ratio, suggesting that the importance of our social interactions, our social networks in the amount of brain that we use on a daily basis, right? Another evidence that suggests that social interactions impact biological fitness comes from studies like these, where people have found that in rhesus macaques, their social rank in their group or social status has an impact on the immune regulation and response to infection. So what was done in this study was scientists artificially manipulated groups in these semi-wildly housed rhesus monkeys, and in such a way that as they introduced, the order in which they introduced monkeys in a group determined not only their rank in that group, the dominance rank, but also the activation of many immune regulation related genes which got activated in a rank sensitive manner, mediated by events like the grooming that these monkeys received from each other, which was, you can see, increasing with their dominance rank, as well as the harassment that they received from higher order monkeys, higher order in their rank, which you can see here as negatively correlated with the dominance rank, right? So this is an indication of, and it's true for even humans, that your social interactions have an impact on biological fitness, as does this data indicate, which is perceived loneliness impacts your mental health as well as physical health, with a recent academic report suggesting that social isolation significantly increased a person's risk of premature death from all causes, a risk that may rival those of smoking, obesity, and physical inactivity. Social isolation was associated with about a 50% increased risk of dementia. So what these facts or data suggest is that our social lives are not just incidental, but they are an important contributor or determinant of our day-to-day well-being. Yes. Yes. Yes, immune response to, I think it was a bacterial. Yes, yes. The response was exactly for, in response to that bacterial antigen, that was the word I was looking for, was differential. Yes, 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 yes. I don't know if that was tested, but potentially could be. But I guess it will be more difficult to have a one-on-one manipulation and response in case of human beings, given that there are so many things that you need to control, which you can do only in model organisms. So, but yes, you raise a right point. The direction is right, that it impacts biological fitness in very measurable, objective ways. Okay. So, yes, like I was concluding, social lives are important for our health. And one of the hallmarks of healthy social living is our ability to connect with others. And typically how that has been studied in literature, uh, an important or model or behavior is empathy, which itself is consisted or thought to consider of two sub-processes. One, emotional contagion, or what is known as experience sharing, which is an observer's tendency to take on sensory, motor, visceral, and affective states they encounter in targets, where targets just mean another social person. So as soon as you see someone in a state, you tend to take on their feelings or physiological responses. An example of that is this, very nicely quoted by Adam Smith in his book, Theory of Moral Sentiments, where he says, the mob, when they are gazing at a dancer on the slack rope, naturally wreathe and twist and balance their own bodies as they see him do, and as they feel that they themselves must do if in his situation. So I hope this is something that you all can identify with, right? So this is considered to be very automatic. The other component of empathy that is very popularly studied is what is known as mentalizing or perspective taking, which is an observer's capacity to draw more explicit inferences about the target's intentions, beliefs, and emotions. And according to the perception and action model of empathy, which sort of combines both these components, and which is conceptualized in this Russian doll model of evolution of empathy, which states that these different components of empathic response are added layer upon layer during evolution, but they remain functionally integrated. So according to this, what happens? Just perceiving a target's emotion and or action seems to activate automatically the corresponding representations of that emotion or action in the observer, which in turn activates the somatic and autonomic responses in the observers. So that is the idea, that how do we end up generating these responses? Because maybe this model is working. And one very popular evidence which is considered in support of this model is what is known as the mirror neuron in the motor theory of social cognition. I don't know how many of you have heard of mirror neurons. They are very popular in science, popular science at least. So mirror neurons were first discovered. These are just neurons that were discovered in the monkey brain first. This is a monkey brain, and they were discovered in a specific region of the monkey brain which was associated with planning of movements, which was the premotor cortex of monkeys. And why were these neurons special? These neurons were found to activate or fire when the monkey picked, say, did any task, say, picked a nut, and not only then, but also when he or she viewed the, or say, the experimenter pick do the same action or pick the peanut in this case, but not when the experimenter was using a plier. So the same action had to be performed while watching it as well as while doing it, the same neurons would fire, right? And they could be multimodal in nature in the sense that they would fire not only when they saw the experimenter break the peanut, which would not only have the visual thing but also the audio input. When they just hear the sound of the peanut being broken, these neurons would fire. And also when, yes, so when they see the experimenter break the peanut, just hears the sound. When they see the experimenter break the peanut and hears the sound, and when the monkey breaks the peanut himself. So that is when the same neuron would fire strongly, right? So according to the motor theory of social cognition, mirror neurons are the mediators or provide an observer with the understanding of a perceived action by means of this motor simulation of the agent's observed movements. But though this was a very popular idea when it was discovered, it has its own limitations. Can you guess what? I've sort of articulated it here, which is first, empathy is not always so automatic as it seems to be by this conceptualization, right? It is often contextual or motivated. For example, it's known that you end up feeling, it's easier for people to be empathetic towards an in-group member versus an out-group member. So it's not so automatic. Another thing is that a specific goals, intentions, emotions, perceptions, desires, etc., may often be far removed from any of their observable behaviors, right? So simulating an agent's movements might be sufficient for understanding their motor intention, but not for understanding these unobservable states. So there is a limitation to this account. So how can one study the physiological correlates of non-observable mental states of others? One approach has been via studying empathy for pain. One very pioneering study by Singer et al. Found that when people were put under a fMRI scanner, which is basically a scanner that can quantify the activity observed in different brain regions associated via the blood oxygenation level, how much blood flows towards one region because it is getting active. That can be used as a metric for quantifying the activity associated with that region. So using these signals, people found that when they were shown signs on a monitor of someone that they loved or their friend or their family member was given pain, those periods were associated with the activity in these two specific regions called ACC, which is anterior cingulate cortex, and anterior insula bilaterally. So these are two regions, ACC and anterior insula, that were found to specifically get activated when people reported feeling bad for someone else's pain. And it, interestingly, not only the activity was higher, this activity correlated with the subjectively reported extent of empathy. So these correlations tell us that not the activity in the brain was not random. It was probably associated with what they reported, right? So because they don't have control over their brain activity that you can otherwise objectively measure. So these correlations were important to tell us that, yeah, people are sensitive to others' state of mind, which is probably not observable in this case. More recently, yes. Yes, good question. So X is the score that was obtained by these questionnaires, which is asking them how is their general tendency for feeling empathy towards people. So in this case, it was again, so you can see here, these are the scales or the name of the questionnaires: empathic concern scale and the balanced emotional empathy scale. So these were the two scales or questionnaires that were used, and people had to respond to few questions, and based on that, based on their ratings, the scores would tell us how empathic one person is as a trait, okay? And those traits were found to correlate with the same people's activity in these two regions. So on the y-axis is the activity associated with ACC, and the lower ones are for anterior insula. Okay, so I think these are beta values. So we were discussing about regression a few minutes back. So these are beta regression factors, so values. So associated with these two particular regions, and they were found to correlate with how much they scored on this questionnaire. Okay. More recently, there have been few studies that have tested the effect of painkiller acetaminophen, which is interestingly the active ingredient of a drug that all of us very happily consume, which is paracetamol, which is considered very safe, of course. So an active ingredient of that drug is acetaminophen, and when people were given a regular dose or a clinical dose for say 4 days or 1 week, they found in these particular studies how not only the activity in the same two regions decreased when people were asked to, okay, so there are two things. One is in multiple studies, they reported having expressing less empathy for others in distress. So they were shown pictures of people in distress in ugly situations, and they reported having less empathy for them following the dose of paracetamol. And the other study is when fMRI was done like before, but it was a social exclusion task. So a social exclusion, a very common task that was used is known as a cyberball task, where you play with agents on the screen, and this is a ball tossing game, okay? So in the first round, you press a button, a ball on the screen goes to the next player or the agent, and like that it is passed around 2 or 3 people. It comes to you after every regular intervals in the first round. But after a second round or so, you stop getting the ball. And it has been associated with people feeling socially excluded. Subjective reports suggest that they feel bad about being excluded from the game. And fMRI activity has shown that specific regions like, again, ACC and anterior insula, which has been associated with the psychological component of pain in the earlier study, activity in these regions go down, increase following a social exclusion task. Yes? What's the drug mechanism of acetaminophen? That's a interesting question. We know that it's not, we know it to a broad extent, not the specifics, but what we know is there are two mechanisms, at least one that goes via the activation of endocannabinoid receptors, which is CB1, which are very rampantly present in the brain. And the second is via serotonergic receptors. So it could be via both also. So we don't know how exactly that connects to these cognitive phenomena, of course, but that is something that we are interested in examining. But there is a limit to how much you can do with human participants, right? So, but yes, there are definitely interesting ways to proceed with that. Yes. They are temporary. I mean, nobody has really tested for longitudinal. I think there's one of the papers did study it after a week or so, but only subjective reports were consistent. Nobody has done fMRI study after a longer period. So yes, so not only acetaminophen reduced the activity associated with the subjective component of pain in these two regions, like you can see here. Yeah, it also reduced the, it reduced the subjective reporting effects as well as the activity in these two specific regions following a dose of acetaminophen. In other studies, people have looked at the role of acetaminophen in reducing even positive actions or reactions to someone else's state of mind. So that is why it's known to be blunting both positive and negative reactions to stimuli, which is interesting when we come to our own results in a few slides. So we looked at these fMRI results and we wanted to test, like pointed out, what are the physiological mechanisms by which this might be happening, or whether it even happens in monkeys, right? So we chose monkeys because monkeys, like previously discussed, they are very close to us evolutionarily and are model organisms that we can actually study using brain recordings, for example. So monkey brains, because they have been a favorite model, have been tested in social tasks before, where we have seen how in different tasks, here when two monkeys are there in the same room and one monkey, which is known as the actor monkey, has to make decisions for the other monkey, how much juice reward, say, the other monkey can receive versus nobody gets any reward. They prefer to give the other monkey the juice reward instead of letting it wasted. Okay, so that is one thing. They like donating reward to conspecifics. That is one result. In another study, people checked if monkeys care if the other monkey gets a negative outcome, which in this case was an air puff close to the eye. So air puff is a sudden blast of air for a small second, but which is very aversive to monkeys. So they don't like even the other monkey getting these blasts of airs, and that is what was found in this study. Okay, so by these results, we knew that monkeys care about conspecifics, not very surprising, but we wanted to, yes, document it anyway, yes. Right. So the way usually it is done is that in a in a session when both monkeys are sitting next to each other, every trial is presented with, say, two options that the monkey can choose from, say by use of pressing a button or moving a joystick, that they associate with an outcome that they can observe right at the end of the trial. So based on that sort of conditioning of what pressing will lead to what outcome, they kind of learn what their consequences of their actions are. And then then you basically run the study after that conditioning is done and see or basically analyze their choices over a period of trials in the session and see if there are any patterns emerging from that. And such behavioral analysis showed these two behaviors in different studies. Hmm. Right. Right. Hmm. Right. Correct, correct. Those are brilliant questions, and actually, some of the trials in that actually tried to tease apart. So, there were trials in which either option would lead to different outcomes. One that would either give the other monkey some reward versus no one. So, that was other versus no one, or it was self versus other. Those trials were also there where you had to choose between self and the other, who gets the reward. And in those, consistently all monkeys preferred themselves to receive the reward over the other monkey, at least in the study that was testing that. Okay.
So were they strange monkeys or were they related to each other?
They may not be related per se, but these monkeys sort of live together in the same colony. So they kind of know or see each other. They have their separate cages in some sense.
Like
But
we can study maternal instincts using this in monkeys.
sure, yes, yeah. Yes, but here specifically they were not related. Okay, so could be friends or they would be higher in their dominance ranking. All those things could also be checked to see if those play a role. But in this specific case it was not tested because they were just a 2 to 3 pairs of monkeys that were tested, because it's a big setup, because you start with behavior, training them, and then you go on to do some neural recordings also, which I will just talk about. So since we know that monkeys are sensitive to other monkeys' outcomes, there have been other studies that have looked at which region in the brain is probably specific to that. And across multiple studies, it was found that a particular region of ACC that we have just seen, anterior cingulate cortex, the gyral portion of it, which is basically, so sulci are the grooves. You can see the brain has these grooves and bulges, right? So the bulges are the gyri and the grooves are the sulci. So that is why ACCG and ACCS stand for, okay? So a particular region in ACC, which is ACC gyrus, was sensitive to all sort of social outcomes, social learning, being sensitive to other rewards, others' rewards and things like that, whereas ACC sulcus was mostly associated with self-related outcomes. And that is what is seen in this sort of data, where 3 particular regions in the brain were recorded from. And when I say recorded, neural recording was done via sending down small wires to the brain area, where you can see and hear the activity of their action potentials or firing rates while they are doing this task, okay? So in trials, when they chose themselves over the other for receiving the reward versus choosing the other monkeys or giving that other monkey reward over none, those kind of different trials, what was the activity of these specific neurons? And they found that if you were to categorize these neurons into 3 types, one that was more sensitive to self trials, one that was more sensitive to others' outcome, that is why it's called other referenced, and some that were equally sensitive to both self and other outcomes, then this was a distribution across these 3 different regions. So OFC is orbitofrontal cortex, which has a maximum proportion of self-referenced neurons, whereas between ACC sulcus and ACC gyrus, the ACC gyrus had a greater proportion of other sensitive neurons. So that is the big takeaway out of it. So we wanted to check whether, going back to the results of the painkiller affecting empathy, whether something like that can be tested in monkeys as well, for which we designed a task which was similar. Monkey can choose to give higher or lower proportion of liquid outcome, which can be sweet or which can be aversive. Now, what is aversive here? It's not air puff anymore. It's a liquid reward, but it's bitter in taste. So it's not really rewarding, it is aversive. So in separate trials, the cue will tell the monkey what sort of positive or negative outcome they can receive at the end. And not only the valence or the pleasantness of the outcome, but also who will receive it. So across 4 different cues, we kind of made them learn this association, that 2 of them means they themselves will be the recipient. One of them will be a good tasting reward and the other will be bad tasting or bitter tasting. The other 2 cues will be for good or bad outcomes for the other monkey. Right, so this is a task. You'll forget the details, but yes, this is again showing the proportion by the shading in the bar, how much of good or bad outcome will the other person receive. So for example, if you go for yellow here, since yellow is the major shaded region in this bar, going for yellow would mean more outcome, good or bad, to the recipient versus going for white where you'll receive less amount, okay? Again, details you can forget if you want. In general, these are examples of 2 outcomes of sessions for 2 different monkeys, and we saw when we were plotting the number of unattempted trials, trials in which the reward cue presents but the monkey does not want to do that trial. Those kind of trials are maximum when the outcome is bad for self and the other, especially in this monkey's case, right? Whereas when the outcome is good for self and other, they ended up attempting it more. That's why there's a lower percentage of unattempted trials, okay? Whereas for this monkey, the a percentage of unattempted trials are in general more for all trials which did not give him good outcome. So it will change from session to session, and therefore we kind of analyze it using regression, again something that we talked about. And here what we found actually, let me go back a little. So this is the percentage of unattempted trials through the course of a session. You can see for self-good trials, it's always sort of close to zero. So you end up attempting all those trials. Whereas for self-bad, it is low in the beginning, but increases as soon as the monkey is satiated. So it does not care for bad outcome anymore. In the beginning, it's thirsty, so he wants to drink whatever you're giving. And the same with other good and other bad trials also. But what was interesting was after a dose of acetaminophen, and after 1 hour, if you record from the monkey again, you see the percentages of these unattempted trials going down. What does it mean? You end up seeing monkeys perform these trials regardless of the valence and who is at the receiving end. And that is what we got from this regression analysis as well, that acetaminophen decreases the unattempted trials regardless of valence, which is consistent with what we found in human studies before, right? No matter what the valence or pleasantness of the outcome was for others, positive or negative, the drug, the painkiller reduced that affective reaction even in humans. So that kind of parallel is interesting. The next thing that we did was, like I was saying, we recorded from neurons in a particular region of ACC gyrus, and we analyzed the slowly changing signal in that brain region. So slowly changing signals are known as local field potentials. This is different from the action potentials you see, which are very fast responses to a stimuli. And we looked at a particular band of or frequency of these slowly changing signals, which was between 8 to 12 hertz, the alpha range, because in human EEG studies, it was found to be sensitive to empathy. Okay, so that's why we chose or focused on this band range. And we found that this LFP alpha power was sensitive to the outcome valence of not only self trials, that is SGSP, but also for other trials. So in some sense, if you just look at the activity of these neurons after the reward was received, whether it be me or the other monkey, these neurons were sensitive to the valence of that outcome, is what this is telling you. And how did we arrive at this? So the y-axis here is the discrimination accuracy. So here the process is something similar to what we were discussing in the previous talk about classification, right? So just an introductory slide on what classification is. It's a machine learning approach to test if there is information inherent in the data, that is substantial difference between groups, that allows it to be classified into known categories with high cross-validation accuracies. Supervised learning techniques in which a mathematical model is allowed to learn from labeled data, that is a training data. So in this case, we give them a few trials for the model to learn which trials positive outcome was received, which trials negative outcome was received, and then you give them test data to see if the model can classify between the two correctly or not. And that accuracy is what is telling us how good the model is in classification. And that is a cross-validation accuracy, a test of how that model is, okay? So there are many multiple ways of classifying. We used something called linear discriminant analysis, which is a very simple case scenario of a classification algorithm. We also use KNN, which is K nearest neighbor algorithm. Later in the talk, if we have time, we'll get into that. But in general, what is LDA or linear discriminant analysis doing? It's basically, if you were to look at say a set of data points, on x-axis, let's say height and weight of two groups of people, okay? And that is what is colored by purple and blue here. We want to find the model should be able to classify these groups apart, right? And what LDA does is to provide this line that can divide the groups into clear, accurately, right? So these two data points in this case are affected by only 2 dimensions, the height and the weight, but it can be used for any data which is multidimensional itself. This is a simple case scenario for 2 dimensions. If the data needs to be classified, what is LDA doing? It is projecting that high dimensional feature space into 1 dimension of a line and dividing the data into 2 groups. And what is the goal or function that it is optimizing while it is drawing a line? 2 things. It is trying to minimize the error between the groups and maximize, within the groups, and maximize the variance between groups. So that's what you can see here. So this, if you draw a line like this, in this example, it is able to increase the variance between groups, but the within group variance is also huge. Whereas if you, okay, let's see, yeah. If you draw a line like this, you can see it reduces the within group variance while maximizing the between group variance. So that is a better model. So you will test that by checking the cross-validation accuracy, but a good model will be able to do both. That's just how what this slide is trying to say. So another method like KNN that I was talking about does it in a slightly different way. Again, it's a classifying algorithm. It makes fewer assumptions. It's more flexible and all that. But what it does is more intuitive actually. So again, for a set of data from say 2 groups, men and women, whose height and weights we have plotted in this 2-dimensional space. We have, again, it's a supervised machine learning algorithm, right? So you give them labeled data sets earlier on. You give them these reds and blues, what data point is from which group. And then you provide a new data set, a star. And then you let the model decide where that star needs to be, like to classify it to the men category or the women category, for example. And what KNN does is that it uses the coordinate space or the distance between this star and its neighboring points and uses that neighborhood space to decide if there are more men or blue dots in that space, this new data set belongs to the men category. If there are more red dots in that neighborhood space, then it should belong to the women category. So that is what it is doing. It's more, yes. Star is a new data point. So suppose your model has learned from the test data that this is how far you will be from the blue group. If you are belonging to that class, you'll be closer to that group, right? So or vice versa. So you decide a neighborhood based on what value of k you want to use. 3, 7, 5 basically decides a number of neighbors that you are basically taking into account. And how in that neighborhood, what is the majority color will decide the belongingness of this new data set. Make sense? Again, probably a detail I don't want to get into. But anyway, so giving you a flavor of what classification does in general. So we were able to find that if you look at the LFP data, after the reward was given, they are sensitive to the outcome valence for both self trials and the other monkeys trials. But interestingly, after acetaminophen was given, this correlation of this accuracy with behavioral valence discrimination, which is behavioral regression factors or regression coefficient, which was there for only other trials, other good versus other bad, that valence discrimination was correlated for only others, and after acetaminophen, that correlation was disrupted. In fact, if you look at the self data, it wasn't there significantly to begin with, and after acetaminophen, it did not change. So what it tells us is that acetaminophen action on LFP of ACC gyrus is specific to other monkeys' outcomes, whether it was aversive or positive. Okay, so this is a nice way of ending this part of my talk. If you have time, I can go on to the next part. But basically, 3 things that it highlights. Acetaminophen reduces the monkey's behavioral sensitivity to the valence of outcomes such that even if it's negative, they are more vigorously doing it. right? So they are less sensitive to the valence of the outcome. I'm sorry. Also that there seems to be shared neural substrates underlying the processing of something like pain and something like negative taste, which is what we used in the task, right? So it's interesting to explore even that, how the psychological components of pain is having overlapping processing substrates with something which is another modality of taste altogether, okay? Any questions so far before I, yes? Huh. Yes. Correct. Good answer. Good question. So that is also actually dependent on how informative your data is. So you typically try different k values and check the cross-validation accuracy of your model, and you choose a model which gives you better results, better classification accuracy with that data. Okay. Okay. Oops. Let's go to the next section, which is connected to the first section in such a way that we talked about similarity in behavioral and physiological responses when we talked about mirror neurons, also about emotional contagion, right? When we are seeing someone being in a particular state, we automatically take that physiological state. Now, that has been explicitly studied under a phenomenon known as interpersonal synchrony, okay? In a recent study, people found that when two people are just sitting across each other, they're not asked to do anything. Either they have been given visual access to each other, or in another condition, they have not. So no, sorry, where is it? Yeah, so this is visual access to each other versus a condition where there was no visual access. And the other condition was they were seated close to each other versus far from each other. So across these four conditions, they had to just spontaneously be there and watch if possible. And they recorded the movements, the spontaneous movements that people do by using these video recordings. So there is a open access software which uses machine learning algorithm based again to quantify the velocity of any body part across frames of video recordings, right? And those movements or movement velocities, which are known as kinematics, was then calculated for each of these different body parts for both people, both persons in a pair. And they found that there was higher correlation between body parts moving with each other in the near visual access condition. All this just to say that, okay, and the other thing is it happens quite fast also, within a second or two. So all this to say that we tend to spontaneously align with people in terms of even small bodily movements, which are probably not something that we are aware of as well. And that is an example of interpersonal movement synchrony, right? Another thing is that we know heart rate is an index of autonomic arousal. So people have used heart rate as a measure to do something interesting. Again, what they have done here is they watch videos of people narrating positive and negative events in their lives, okay? While there is heart rate being monitored for not only the person who is in the recording, but also the participant who is viewing these recordings. So while the person is viewing these recordings, they are also asked to rate the specific emotions that this person might be going through. For example, how much anger, disgust, what is that, embarrassment, excitement, fear. So these are special emotion categories that the participant is asked to rate on for the person who is narrating life events, okay? And what they found was, again, to cut the story short, they found there was increased heart rate synchrony associated with accuracy of identifying the emotion in the other person. So how do they know whether the emotion categorization is accurate? They ask the people separately who are in the video to rate themselves as well. So if there is a match between what they rated while they viewed their own recording versus the participants guess of what the other person's emotional state would be. That is considered as high empathic accuracy. And high empathic accuracy for these specific emotions was correlated with high heart rate synchrony, suggesting that heart rate synchrony is associated with our guessing what is someone else's emotional state. Okay? Again, there have been different measures of quantifying heart rate synchrony. In the earlier two studies, synchrony itself was measured by just correlations. Here they use something more sophisticated, which is sensitive to the nonlinear dynamics of heart rates as well, right? So this is a actual field study in some sense. In a small village in Spain, there is a fire walking ritual. So they recorded the activity of the fire walker, the heart rate activity of the fire walker, and some of the people in the audience as the fire walker was going through that patch of fire. In the audience were people who were close to these fire walker, like spouses or family members, and then there were of course other strangers that they recorded the heart rates from. And they found, and these are example heart rate signals of, let's see, so blue is for the fire walker, red is the related spectator, which is the wife in this case, and this is a non-related spectator, right? And as you can see, at least in this very illustrative example, there is more synchrony between the fire walker and the related spectator as opposed to with the unrelated spectator, right? So why I say this is a more refined analysis of synchrony, it is known as recurrence quantification analysis because it is sensitive to the temporal order of the dynamics of the heart rate signal. I don't want to get into a lot of it, but maybe a small flavor of it. Yes, if there is time. Mm. 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 Oh. Mm-hmm. Exactly. Right, right. Yeah, so. Excellent question. We don't know yet, but frankly, we are getting there. So people have used across multiple studies, just looked at brain signals between people. For example, they have used EEG, which gives electrical signals. They have used people in the scanner, that is fMRI scanner, looked at the bold or the oxygen level dependent signal, and they found that the more there is similarity in how they understand a particular phenomenon, more is the similarity in the brain activity of these people. So for example, there was actually a recent study where a bunch of people were asked to ask their opinion after watching a movie clip, and initially they had different opinions, but following a discussion which was meant to increase consensus, the increase in consensus was correlated with increased similarity in brain activity after the discussion. So, and there have been other studies showing that shared understanding of a particular story or a narration is associated with similar brain activity in the people. And heart rate is been tested in different contexts, like this is one, and the one that, let me just skip through all this. To go to our study where we looked at heart rate synchrony for something as naturalistic as group decisions. Okay, so again here our interest in looking at this was from the idea of what is known as groupthink, which probably you might have heard of, which is a tendency for groups to think in harmony. Though that looks like a good thing, that may not always be the case, right? Because when you are put together as a group, you are expected to add to the discussion in unique ways that probably one person could not. So that's why your groups are supposed to be better at problem solving, because they can each provide their own expertise and unique perspectives. But sometimes when people don't do that in a group, you can end up with inferior outcomes. And that is what groupthink is called, which probably is inspired by or motivated by the desire to maintain harmony and cohesion, but that may not always be a positive outcome. Because, and the other thing is that members may seek to avoid conflict, feel uncomfortable expressing opinions that are different from others, for fear of disapproval, negative evaluation, or even dismissal from the group. So this is something what has been characterized in organizational behavior literature as team psychological safety. So if you feel safe in a group to express yourself with an opinion that is different from the others, then you have high psychological safety. So we wanted to test in a problem solving task whether heart rate synchrony between members of a group is associated with productive outcomes, whether they were able to solve the question correctly or not. And we used, again, heart rate measures, baseline during the group discussion, and also post-task survey, asking people how satisfied they were with the group discussion that they had. And we went through this analysis that I did not get into a lot of detail of, but it's again a multi-dimensional recurrence quantification analysis, because we use data from now more than 2 people. Actually, these groups were made of 3 to 6 individuals. And they could either reach the correct consensus answer or they did not. And we found that their heart rate measures or heart rate synchrony measures were able to pick out or classify which groups would be productive versus not, with 70% cross-validation accuracy. So it's again a classification result. And it was significantly higher than either self-report assessment of group function, as well as the baseline group measure of heart rate. So this suggests that heart rate synchrony during a naturalistic group discussion task could be a biomarker of effective information processing in collective decision making. Now, how that relates to brain signals, which people have independently shown to also synchronize when you understand something in the same way, is a future question to look at. That's all. These are my prime collaborators for the last study, which we recently published. My postdoc advisor and my students, Swarag and Nivedita. And this is our lab currently. Feel free to ask any questions now or by email. I know I probably took more time. Thank you.
Questions?
We can always chat later. I'm here tonight, so let's meet over dinner or later.
If not, then let's thank the speaker for this interesting talk.